Welcome to NOAA Live Alaska. My name is Emma Radish, and I'm going to be moderating today's webinar. This series is a collaborative effort by NOAA Fisheries Alaska Fisheries Science Center, where I'm interning, NOAA Alaska's Regional Collaboration Network, and NOAA's National Weather Service. I also would like to introduce my supervisors, Lisa Haruki-Raring, Story Oates, and Mabel Baldwin-Schaefer, as well as my co-intern, Elizabeth Sajkowski. They'll be helping me behind the scenes today. This webinar series is designed to help you get to know NOAA's work in Alaska, our partners, and the communities we work with in Alaska, and how we connect with your communities. NOAA, the National Oceanic and Atmospheric Administration, studies the ocean, atmosphere, and where the two meet, from the weather and to the animals that live around us. This school year, we're presenting a short series of webinars that are regionally based so that we can focus on topics of interest to specific areas of Alaska. In addition to work from NOAA scientists, we are also aiming to present perspectives from the communities we work with, and research conducted by students. We hope this gives you a look at different communities as well as different career paths you might be interested in. Today we're starting our webinar series for the Bering Strait with a presentation by Emily Markowitz, who works at the NOAA Fisheries Alaska Fisheries Science Center. While we will be talking about NOAA's and partners' role in research and stewardship, we want to recognize that we are all coming to you from the traditional lands of Indigenous communities who have substantial traditional and local knowledge and much to share with us. Emily's work is conducted in the Bering Sea and Gulf of Alaska, which from north to south include the traditional home waters of the Inupiat, Yupik, Siberian Yupik, Ununuk, Aleut, Eyak, Tlingit, Haida, and Tsimshian people. We would also like to acknowledge that Emily is presenting and we are hosting this webinar from the traditional lands of the Coast Salish, including the first people of Seattle, the Duwamish people, past and present. And now just a few guidelines before I hand you over to our speaker. You are all muted because we have a lot of people on the line and want to make sure everyone can hear Emily. However, there is a box where you can write questions. We encourage you to ask them as we go. Our behind the scenes team will be keeping track for Emily, and she'll stop every now and then to answer some of the questions. We may not get to all of the questions, but we'll try to answer as many as we can. All right, now I'll hand it over to Emily to introduce herself.
Hey everyone, it's so great to meet you all. What an exciting opportunity to share some of the cool stuff I get to do as a fisheries researcher for Alaska Fisheries Science Center. Yeah, I'm excited to hear your questions and share with you some of the exciting things I get to do. A little bit about me. I am originally from New York. That's me as a little kid at the Coney Island Aquarium, or not, maybe. That's me at the Coney Island Aquarium as a kid. I loved going to the aquarium. I loved seeing the scuba divers in there, cleaning the tanks. I just thought that was the coolest thing. After, going through K through 12, I went to Stony Brook University, their School of Marine Atmospheric Sciences. I got a undergraduate degree in marine sciences and a graduate degree, my master's in marine sciences as well, focusing on fisheries science. At the end of that, I had the really amazing opportunity to make my way down to Washington, D.C., where I worked for NOAA as a fellow in the John A. Knaus Marine Policy Fellowship, actually working with some of our favorite charismatic megafauna. So that's a funny, cool way of saying protected species, or whales and dolphins, coral reefs, seals and sea lions, loads of other species that we take a lot of special care in protecting. After my fellowship, I actually got to work on issues that, on understanding issues that are affecting fishermen and how much fish they catch and they get for that catch money-wise, the decisions that they make when catching those fish, and what fish they want to catch. So that was with what we call the socioeconomics group. I am so lucky to have been applying to some jobs, and I got this really amazing gig where I am now at the Alaska Fisheries Science Center here in Seattle, Washington. But every summer, of course, I come to visit your backyard in the Bering Sea. So it has just been really amazing to delve deeper into this incredible environment, an ecosystem, and learn from not just the fish and the water that's there, but also from all of you. And I look forward to also learning more. So little bit more about these surveys, right? There's actually 2 different surveys that we do. There's actually many surveys that we do, but the Bering Sea team focuses on 2. There's the Northern Bering Sea, which is surveyed intermittently, and we're starting to do that a lot more regularly. In terms of the standard survey, what we would use in our data analyses, we've done it 4 times, and it's funding limited, but we're making a good push to do it maybe more regularly in upcoming years. We'll be there in 2022, this year. And then also, of course, our famous Eastern Bering Sea survey, which we've surveyed annually since 1982. Obviously, we didn't get to visit in 2020 because of COVID, but otherwise it is a really consistent, powerful data set, and there's almost none like it. And we survey these areas to understand more about not just what fish are there, but how many of them are there, what the wellness of those fish are in those populations, in those ecosystems. We also take environmental data too. So just to give a little bit more sense about like how does one do a survey, right? We have all these stations, which here are represented by these grids, and then we have boats that go through and actually go to each of those stations, collect fish and different environmental variables. And we do this in a very systematic design. So you can see we're moving from east to west when we're going up and down. And we try to do this consistently for all of our survey years. So we can get a better sense of how things are changing over time and place. And so you can see there's an A and a V, that's for the Alaska Knight and the Vester Allen. Those are the 2 boats we used last year. And the colors that you're seeing appear as the boats go to each of these stations are different bottom temperatures that we collected at each of these places. So we're not just collecting those fish, right? We're of course also collecting these bottom temperatures. And we're doing this from about May to August every year. Now, you know, being on the boat is the coolest. Can't say that enough. You know, I was in Alaska for the longest day of the year, and it was just twilight. I had never experienced that being from New York. And on other trips, it was amazing to see. We got some beautiful days. You can see in the lower right-hand corner where my friend Erin is reading a book on deck on this beautiful warm day. We also get some weather days, which are still super cool, and that's in the middle upper photo. We're waving to our friends over on the Alaska Knight as they pass by us. We're just hanging out, waiting up for the waves to pass. But it's also great because you make some really great friends and you get to celebrate your hard work and achievements. You know, only what you have on the boat is what you get to use on the boat. So you don't get to go to the grocery store and pick out new ice cream or pick up a favorite food that you like. So you get to celebrate harder days by maybe making some baked goods. Here I made some bread, and those are it's just a fun recipe, and it takes a really long time, but everyone really enjoys it. And you get to tell people that you really appreciate them and their hard work. I made this thank you card when I left the boat. We have shab is this all this extra stuff that you collect that isn't easy to parse apart. So I told them it was really shabtastic working with everyone. And I have a quick video to give you sort of an image of what it means to work on the boat. See if I can, yes, perfect, Lisa. You got it?
Okay, so I'm just setting up the video now, and remember that it'll reset, or it'll mute you once it starts. So if you're going to talk, you remember to unmute yourself.
Yeah, excellent. You guys can hear me?
Yes.
So what you'll see here is that we have this net of fish that we pulled up, and we're dumping it into this sorting table so then we can go through with all these baskets that you can kind of see for free in the sides of this photo, and start sorting them so we can understand how many fish there are and how big they are, length and weight, and do some other sampling. And so that's kind of what it looks like in its initial state. And after this, we take all these fish and we do some other studies on them. And
Okay,
get
so
back to my
made you
screen.
presenter
Sorry?
again. I just made you the presenter again.
Perfect. Yes, you did. And you guys can see my screen. Cool. So, you might be thinking, why would we take fish to study fish for fisheries, right? Well, and how do we make the decision to take those fish? So we call it a sample from a population, as you can see in this diagram here, right? We want to get a best understanding of the population, so we take a small sample and we study that. And here there are all these different colored people, and so we take a representative sample. We're taking one of each color, or try to. Maybe we miss green, but we're trying to also get green too. And so there's a lot of research into how to take a useful sample from a population. You might think about it like, if I wanted to know what the favorite color of people at your school was, let's say the most favorite color of all the students who went to a school, I could sample one classroom, let's say a third grade classroom, just one, and ask everyone there what their favorite color is. And it might tell me about what the favorite color of most people at the school is, but probably better to get, take half of a third grade class, half of a fifth grade class, maybe half of a first grade class, because then I'd start to get some diversity in who I was sampling from the school population. So if you think about it like that, we go to each of these stations and we put a net in and we take fish and sample them the same way at each place, so we can understand how the fish population is across space and time, but we just take a few fish to ask that question. So I talk about how awesome being on the boat is. Well, some days are a little harder than others. You can see me in the upper right-hand corner, hugging some gear that I'd really like to just work easily, because I'm very tired, and I'm very tired of it not really working with me all that nicely. The lower right-hand corner has a bunch of sea stars, purple orange sea stars, and they're really cool, and I still think they're really cool, but they are really good at sticking together, so they can be very hard to pull apart and to count. And then sometimes you have the misfortune of pulling up an old crab pot, right? It's not someone's fishing with that crab pot. It's lost its buoy, and so it's been lost. And so it's just collecting all of these critters, and we call it ghost gear because it will continue to fish even though it's not in use, so to speak. And so we have to keep those on deck and break them down, and that can take a lot of time from the survey. So yeah, some days are a little harder than others, but it's all in this really amazing experience that we get to be part of. So I'm going to take a little bit of a break here. I've introduced a lot of different things. I want to know if you guys have some questions.
Yeah, we have a few questions in the chat.
Wonderful.
Go all the way back to your bottom trawl surveys. We're wondering how the bottom temperatures of the water can be below freezing when it's water.
That is an excellent question. Well, so fresh water would freeze at 32 degrees Fahrenheit or 0 degrees Celsius, right? This is salt water. It's very different. And so it actually will freeze at a much lower temperature, about 28.4 degrees Fahrenheit. Also, when you're that deep, pressure can affect temperature, and there's this whole fun equation that kind of dictates that, but it gets into some sort of chemistry and physics. Moral of the story is it's salt water and it has a different freezing point, and that's why you can get below freezing temperatures.
Awesome, that makes a lot of sense. And then we have a question from Theodore, who's asking if you sample only fish or if you sample also other animals like dolphins and sharks.
That is an excellent question. I'll get a little bit into that in the next slides. We don't catch dolphin, or haven't, so that hasn't come up, thankfully, because that would be, actually it gets really complicated when you catch a protected species, larger and The word I'm looking for is, well, there are different regulations and protections on say dolphins. We'd love to catch more sharks because they're the coolest, but our survey doesn't typically catch sharks. We do catch a lot of scapes. And yeah, right, so to really answer the heart of your question, we catch not just fish, but we also catch invertebrates, we catch a wide array of fish. I'll show you some pictures of that next.
Great. And then we have another question from Miss Duchesne's 3rd grade class, and they're wondering, how do you actually fish on the boat? How long does it take, or how much time a day? What kinds of tools do you use?
That is an awesome question. Yeah, so we, the show starts when we put the net into the water, and then we leave the net in the water for about 30 minutes, and we see what fish we catch, and then we bring that net back on deck, and then as you saw in that video, we take the net and we dump it into that sorting table in the middle. And then we have a bunch of people around the table, and we have those baskets on the ground, and so we're sorting all those fish into those different baskets. We then take those over, and you can see it actually in the picture here. Can you see my cursor? There's a scale here, and so we'll weigh all those baskets, and then we'll take another subsample of those fish and we'll length them with a ruler, or fancier ruler. They're these big old length boards. You can see them over here in the lower right-hand corner. And then we do a couple of other samples that we'll talk about in a few other slides.
And Emily, is that the net that's wound up on the spool at the very back of the ship?
Sure is, on the A-frame, because it looks like an A. Yeah, so the net, when we're not using it, we pull that back up onto the spool, keep it out of the way. And you can see over where this person's standing in the sort of right middle of the photo, there are some backup nets, because sometimes you rip them and you can't get back to shore that quickly. So you have to have some extras on hand.
Awesome. And then we have another question from Dawn Davis, and they're asking, how many pounds of fish a day do you net?
Well, that varies. The Northern Bering Sea, we catch a lot less fish in a tow than we might in the Eastern Bering Sea. And we might not do the same number of tows every day. So probably better, if I could reframe your question, if you don't mind, how many pounds of fish or how much weight of fish do we catch in a haul? It can vary. Let's say the smallest could be like couple hundred kilogram of fish, kilos of fish, but really big ones can be, I would say a really, really big one that's like typical, maybe 4,000 kilos. But it all depends on where we are and what the net catches.
Awesome. And then one last question before we let you move on, Em. You were talking about all the different types of fish you catch and how cold the water is, and people in the chat are wondering, how can fish live in such cold waters?
Yeah, oh, it's an awesome question. So some fish, like the Arctic cod, which some of you may be really familiar with, actually have these antifreeze proteins. So basically the proteins bind to these tiny ice crystals in their bodies, and it allows them to exist in these really cold temperatures. And there's all this really cool research behind it, but I'll leave that to someone who actually studies Arctic cod. But we call them antifreeze proteins. They're adapted for this particular environment, which is just so cool.
Awesome. Thanks, Em. Why don't you move on to your next section?
Totally. Yeah. So now you get to see some of the cool critters that we catch in the survey. Let's see. Yeah. So, you know, I grew up on the East Coast, and I had only read about so many of these species, and finally being able to see them is just really incredible. Here I'll go in sort of clockwise. On the upper right-hand corner, we've got a Bering wolffish, and they've got these gnarly teeth, and they're just so wild, and they sort of snake around. We also have our friend, the walleye pollock, which is so incredibly important to the economy of Alaska. It's my friend Joletta holding it up for the camera. We also catch blue king crab, right? So they're not a fish. This survey is really important for understanding how that population for blue king crabs, red king crabs, snow crabs more are doing. Our friend from a couple slides ago, the purple orange sea star, there's actually a lot of interesting research being done on them and how their distribution is changing. Actually, on a lot of these species, but I've read up on the purple orange sea star a fair bit. And then I just think these are like the most beautiful fish. They're called starry flounders. And so they've got these little bumps on them and these colored fins, but they're flatfish. And so they're really, really neat. And there's just so many more, right? I could go on and on, right? We've got these beautiful, like, I want to call them sweater starfish because they remind me of Christmas sweaters. But they're adorable. They're smaller than my hand, right? So let's say they're about the palm of my hand. Then we've got this splendid hermit crab, and it's splendid because if you kind of see it on the claw, they're a little effervescent, right? There's this pink purple that's not really a solid color, right? So we call them splendid hermit crabs. One of my personal favorites is this sturgeon poacher. Sturgeon are my favorite fish. Growing up in New York, there's the Hudson River, and the Hudson River has Hudson sturgeon, and there's a whole story about that. But anything named with sturgeon, I just think are the most amazing. And they look like a sturgeon too. They've got the nose and the ridge back. So they're super cute. Below that, I won't make you guess. This is called a snailfish. And they're kind of this gelatinous, funky fish that lives at the bottom of on the bottom of the ocean. And I just think that they are so wildly cool. I'd never seen one before, but I'd read about them and I think they're fabulous. And then maybe if you guys have gone fishing up in Alaska, you probably caught a yellow Irish lord, which is here on the bottom, on the lower left bottom. I'll show you a quick video of this Telmesis crab. He's just super cute. Actually, Lisa will show you the super cute video of this Telmesis crab because they're alive. It is an honor to get to research them and work with them. I don't know if you could see it, but he's bubbling up. He's trying to let me know that he doesn't appreciate me getting in his face. It's okay. He went back to the ocean and hopefully lives a very happy, long life there. You guys can see my screen again? Yeah, I think you can. We also got to see a whale. We were not surveying anymore. It was the end of the day and it just came up right by the boat. Beautiful whale, right? You can see my friend Joletta again. She's sorting through different snow crabs, taking length and just different measurements on these crabs. And the top is a baby red king crab. They're so cute. Being from the East Coast, I had only ever read about salmon, and so here was a salmon in the flesh. I was so excited to see it. It's just incredible. So much of the world's economy is based off these salmon, and I have had now the privilege to meet one in person. So you can see that I'm pretty stoked about that. I know this might be so, every day for you guys, but for me, I just ecstatic. We saw octopuses and huge skates. That's my friend Rebecca. So when there are skates that are too big for our length boards, we'll set up a person. We know how tall the person is. So we can kind of measure how tall that skate is and we can verify that in our data. If someone's like, wow, you caught a skate that big? It's like, yes. And we took a picture of it for reference. And sometimes you just got to use what you got. And sometimes what you got is you. So that's what Rebecca's doing there, taking a picture with this beautiful skate. And then we catch a lot of invertebrates, shelled invertebrates and beautiful shells and critters. And obviously they go back into the ocean once we count and identify them. So, here are all these incredible species that we get to catch in the survey to understand about this ecosystem. Why is it important for us to study all the species in an ecosystem? Why not just the commercially valuable ones? And I'd love to know what you guys think. I have an answer, but I bet you guys have one too.
So for all of you guys in the audience, if you have an answer to Emily's question about why is it important for us to study all the species in the ecosystem, go ahead and put it in the chat.
We've got a few answers coming in. It looks like Theodore thinks because of climate change. Pascal thinks they might become valuable, all the different species, and that we need to help all of the species because of climate change. Alice and Paul think because other species eat them. Jasper is saying because it is important to know population levels. These are really great answers, guys.
These are awesome answers. You guys are so smart. Yeah, you know all this already. Yeah.
And Grace is thinking maybe to see how the species in the overall ecosystem are doing.
Yeah, these are top-notch questions, top-notch answers, you guys. Understanding how the entire ecosystem is doing is really an important clue into how specific species in that ecosystem are doing. So absolutely, and then we can take that. We have this incredibly long time series. 1982 is an amazing treasure trove of data, and that can help us understand how these different critters have moved over time because of changing water temperatures. We'll get into that a little bit next, but you guys got it on nail on the head. That was awesome. It's not just about the commercially valuable species, and right, of course, who knows? Maybe some of these species will become valuable as for commercial fisheries later on too. We've definitely seen that around the world, if not also, of course, in Alaska. And you know what I think is really a good visual here is like everyone in an ecosystem has a part to play, right? If you live on the bottom and you're a crab or a flatfish or a shellfish, right? Your food, or you eat things from above that fall from the top, or create different environments for other fish. Let's say, you know, you dig up sand from the bottom and you make these sandy areas as one critter, and that's important for another critter to lay their eggs. Phytoplankton, which are in the middle in the green, they're incredibly important as a food resource across the ocean. Yeah, right. It's like everyone in an ecosystem has a part to play, and it's all interconnected. I'll take a few questions about these incredible species. If you guys have them, I'd love to hear.
Yeah, that's great to hear all about the ecosystem you're working with, Em. And we have a few questions relating back to the boat. We even have someone on, Jane, who crewed on the Vesteralen for GOA bottom trawl survey in 1996. So we've got a lot of people who want to hear more about the boat. And one question from Beth is, how long do you stay on the boat at a time?
That's an awesome question, Beth. Also, so exciting that you were on the survey before on the Vesteralen. That's the boat I was on this summer. So yes, okay. So we do 3-week legs, which is so we do 4 legs. We go out it's 4 teams on 2 boats, but there's like 8 different sections of the survey happening. And we do 3 weeks per leg. So this past summer, because of COVID, you had to, the original plan was you had to quarantine before getting on the boat 2 weeks. It changed over the course of the year, but so we ended up not wanting to quarantine for 2 weeks each time. We go for 2 legs. So we did 6 straight weeks on the boat this summer, many of us.
Awesome. And then a little bit more about the fish that you guys are taking surveys of. Roxy's class would like to know, what do you do with all the fish and if you eat any of them?
That is a great question. And so we mostly put them all back in the water, and a lot of them, some of them survive. especially the hardier species, the invertebrates. But a lot of them we take other samples on, which we'll talk about in a few other slides. We sometimes eat them, but only if they're really gonna die, and sort of a sad case. But really, mostly we don't.
Great. And then another question from Roxy's class is, what is the most common fish that you guys catch?
Really depends on where you are, right? Like I showed a picture earlier of a tow that was entirely made up of purple orange sea stars. That doesn't always happen, but you can get tows that are like that. Often it's a mix of different species. Some are much bigger than others, so they seem disproportionately like they make up the majority of the catch, but really there's just a few of them and they're just very big. But we'll catch Pacific cod and walleye pollock and a lot of yellowfin sole and Alaska plaice, evertooth flounder. Yeah, it's a huge list. You can catch a lot of really cool species. And of course, a lot of shad, which is a lot of that broken up shell and some rock. And of course, a lot of little invertebrate critters like different mollusks and little crabs and stuff like that.
Awesome. And then also, a few people were wondering if you see marine mammals like dolphins and sharks, and then why you were interested in studying the fisheries instead of those types of mammals.
Yeah, this part of the ocean, we don't see a lot of sharks and we don't see a lot of whales and dolphins. That was the one whale that we saw that I had that little video clip of, which is like so cool, but it's like the only one I saw. And we go by some islands where there are definitely seals, but we don't see them so much. So yeah, we don't see them as much as we might in other places. Why do I study fish over those cool guys? Well, I'm a sucker for a silver kind of gross looking fish. I just think they're the coolest. So and they have some amazing adaptations for their environment. So do a lot of protected species like dolphins and whales and so forth. But I don't know, these guys kind of have my heart. I just think they're really neat.
That's awesome. And then another question from Mabel and Ruby. They're wondering, since all of your studies are in Alaska, why are you based in Seattle and why are so many other scientists based in Seattle?
That is a great question. I feel like that goes back to some historical decision that got made a long time ago. But I think Seattle is often referred to as the gateway to the Pacific Northwest, to Alaska. And so I think it's a way of keeping Alaska connected to the continental US and not letting people forget how important it is.
So Emily, this is Lisa from behind the scenes. I think a lot of the Alaska fishery is based in Seattle. And so I think that was one of the reasons why the
That's
Alaska
a much better way of saying it. I like that. Yeah.
Yes. But we
Thank
do
you,
have
guys.
we do have offices up in Alaska
Many.
for Mabel and Ruby's question. So there are many of our scientists who are up in Alaska.
Many. I'm based in Seattle, but I have lots of colleagues who are based throughout Alaska.
Awesome, great. Thanks, Lisa and Emily. And then looking more about the ecosystem you were talking about, moving away from the boat questions, Grace noticed that you were talking a lot about phytoplankton, and they were wondering, do you do any observations of the phytoplankton present in the waters that you're collecting larger survey samples in?
We don't. I think that would be a really cool thing to include. There are other surveys that do. Yeah, no, and I've been on surveys on the East Coast that do, and I just think they're the most incredible critters. It's great. But yeah, not our surveys.
One of the things, though, that you did mention, Emily, was that that you take water temperatures, and those were the colors that were on your map. And I think that's of a lot of interest to people in Alaska communities to see what the surface water temperature is on your surveys.
Yeah, that was a map of bottom temperature, but surface temperatures are also really interesting too. Yeah, I totally agree. And we get that data as well.
Great, and we're just going to take 2 more questions before we let you move on to your next section. And one of them was from Allison Paul Wondering if you have nets that keep you from catching the different things you don't want to catch.
So for this survey, we want to get a representative sample of everything that's there. So it's a very general net. There are some decisions that are made to not catch really small things. So you can do that by how big the holes in the net are. So if they're really tiny, you'll catch really tiny things in the net because they'll be able to escape through the holes. And if you have bigger holes in the net, then you will be able to let those little things through. So we call that selectivity. And we don't make a lot of selectivity choices in our net, but we do let the little guys through.
Great. And then just wrapping up this section of questions with a question from Dawn's class. They're wondering, what was the scariest moment you had on the boat? Maybe it was bad weather, something else?
I don't think I had a scary moment on the boat. It's pretty, I've been on boats before and on surveys. Sometimes it gets rocky. I think that's fun. Sometimes when it's rocky and you're working on deck, you always have to be very careful to be mindful of where the net is and where other heavy things or things under tension are. So I don't think there was a time that I was scared, but it is very important to be respectful of the situation that the work that you're doing and the process it takes to do that work, and to put safety first, because then there would be something to be scared about for sure.
Awesome. And we'll get to some of those other questions in the chat later, and we'll let Emily continue on with her presentation.
Cool. Let's go click here. So if I haven't sort of gotten to it, said enough, right? Why do we, that's weird. Why do we catch and count fish? Did you know that Alaska, just the state of Alaska, has commercial fisheries that account for about 60% of the United States' catch landings? So the number of fish that are actually caught and part of the United States' total amount that is fished, and about 1/3 of the United States' revenue. So it's one state of 50 accounts for a massive proportion of the commercial fishing industry in the United States. That's Alaska. It's amazing. So you want to do a really good job of understanding the system. And Finn's curious, that's from the Fisheries Economics of the U.S. report. Back when I was in DC, I actually helped produce that report. And so there's a link to it below if you guys want to check it out. So we kind of talked about this a little bit before, right? We want to know if there's species on the move. We want to know where they are, how many of them there are, and if there are changes over time. And is that because of environmental variables? Is that just natural oscillation of population? Excellent questions. Here, right, you can see that this is walleye pollock, and we have this simple map that's showing where these fish are in higher abundances than in other places. Green is like very low but consistent, right? There's only a very few places where there's no catch. That's the gray. And then the darker the color, the more fish there are. We also want to know, right, so how that correlates with, say, temperature. So here we have a map of bottom temperature from the Bering Sea for all these different years, 2010, 2017, 2019, 2021. And we want to ask, is there a relationship between the really cold temperatures and where these fish are? And here, anything less than 2 degrees is considered the cold pool, which is a variable that's really important and becoming even more important as we study how important temperature is to these fish species. We use it as an index, a way of sort of simplifying a greater phenomenon, and it's not really phenomena, but trend in temperatures in the Bering Sea. And so here it's that blue, darker blue, and really dark blue that is that cold pool, less than 2 degrees Celsius. And what you might notice, right, so there's the cold pool, I'm showing it out here. The cold pool has changed in size. Each of these years it's just a little bit different. And one thing you might notice is that in years where there's really big cold pool, we might see walleye pollock on the edge of that cold pool, right? In years when it's really small, there's a lot more walleye pollock. And that just happens to be with the species. And other species, they need the cold pool. Those temperatures, those low temperatures are really important for them. And so you'll see more fish species when there's larger cold pools. Here, they're obviously avoiding that 2-degree line. And that's just what happens with this particular species, but that can be really informative for understanding what impact, say, this particular variable temperature has on where these fish are. We also gather information about fish age structure. So that's a funny way of saying we take all the lengths of these fish and then we try and understand how old those fish are. And then understanding that over time can be really important for supporting sustainable fisheries, which is what NOAA is all about, sustainable fisheries. Here we've got these little flounder on a length board, and we're trying to tell how big they are, and that goes into a diagram like this where we understand how many fish there are at these different lengths, and if they're male or female or unsexed. And we do so much more, right? There are special projects that look at the genetics of these fish that we catch to see if they're part of different populations of fish that they've developed separately for a while. And you can see that in their DNA. There's stomach analysis and food web dynamics, right? So we can look at the contents of different fish in their stomachs and see what kind of fish they're eating, what fish they're relying on for primary food sources. And then talked about the cold pool, but we also try to assess how this might interweave with seasonal ice cover and the changes that that causes and what fish are around or not around. I have a lot more information on these kinds of special projects, but I think I might take a moment here and just see if you guys have any questions.
Yeah, we still have those 2 questions from the last time. So Theodore is wondering if there's a kind of net that keeps whales, sharks, and dolphins and other bigger mammals out. I know you said there aren't a bunch of them in the area you were in, but just if there were.
Yes. Kind of depends on place and some of the behavior of these species. But they have a name, and the name is escaping me at the moment, but a very similar net is called a TED. It's a turtle exclusion device, right? So you add it to a net. So then if you were to catch, say, sea turtles, they would be able to escape. There are similar models for other species. So yeah, if you look up a turtle exclusion device, it'll kind of give you a sense of what that looks like. And yeah, they definitely exist.
Great. And then we have a question from Roxy's class that's really interesting. And they're wondering what kinds of patterns you're seeing in fish population numbers from your survey. They're thinking about the Noatak River, where they are, and how some years there's more salmon than other years, and wondering if your survey helps explain these trends.
That is a great question. So this survey, I have a picture of a salmon, but it's one of, I think, 3 salmon that we caught while I was on the boat. So we don't catch a lot of salmon, and this survey probably isn't super helpful for informing how salmon populations are doing, but there are slews of other groups, both part of NOAA and elsewhere, that look into those specific trends. It really depends on the fish. Some fish, right, they will avoid really cold temperatures, so when there's not a lot of cold temperatures, they have more space to be part of that ecosystem and to have a wider distribution over that space, like I was talking with the walleye pollock. But there are other species that, like Arctic cod, they have this antifreeze protein, and it's expensive for them to use that and to create that protein. So they are going to find places to use it. So they want to live in really cold places. And so when there's not a lot of cold temperatures, those populations don't do so well. So if we look at trends in temperature, we can start to surmise some general what probably might be happening to these fish populations, and then we use the word ground truth. We can test that in parallel with how many fish we're catching and where they are when we catch them.
Great answer. And then Mr. Shane's class was also wondering, how do you know how old the fish are that you're checking the ages for?
That is an awesome question. I have a slide about that coming up. They have these bones, we call them ear bones or otoliths, and so you can count the rings on them, and that will tell you how old the fish is. And those rings are develop because of seasonality, right? So it gets cold and warm and cold and warm, and so those rings develop at each season. Different for different fish, but it's generally true. And yeah, I'll show you a little bit more about that next.
Great. And then just a few more questions. Grace is wondering how you pick which individuals in your bigger survey to do measurements on.
We try to do it randomly, which can be hard sometimes. You know, if you only catch five fish of a fish that you're doing extra sampling on, well, then you would take all 5 of those fish. If you catch 500, well, I don't want to length and yeah, I don't want to length 500 fish. So I would try and take a random, like, so let's say that makes 100 fish basket, there's 5 baskets, and I only want to take 2 baskets. Well, then I might try and come up with a randomized way to select those baskets. Randomizing can be tricky, but there's a lot of different ways to do it.
Awesome. Yeah, and another great question from Allison Paul. They're wondering, is the water column like the atmosphere, as an example, where the coldest part isn't necessarily at the bottom, or is it always that the bottom of the ocean is the coldest?
That is an awesome question. And I want to give a really complicated answer, which is not that helpful. But yeah, right. There are different layers of the ocean in the water column, and they are driven, and these different sections of the water column are often driven by different, like, other characteristics, say light. And it really depends on how deep the water is. So yeah, I mean, it really depends, but I would say typically the bottom is pretty chilly, but that's not like a general truth. But yes, that would be cold. I don't know. Sorry, I feel like I'm fumbling this question, but there are a lot of different sections to the water column.
Yeah, awesome. And it's great to see you're up here, Emily, sharing your work as a woman in science. And Mabel and Ruby were wondering if you have any guess about the percentage of fishery scientists that are women, or Alaska commercial fisher people that are women.
I don't have stats on that, but yeah, awesome. Thank you. Yeah, it's awesome to be here. One of my last slides, I've got a picture of the team I work with, and you'll get a sense for who's on it. But I would say I get to work with a lot of amazing people, men and women, and I guess our group's really 50-50, which is awesome. It's great to be in a group that word I'm looking for is yeah, has this really diversified approach to working through issues, and it's really inclusive. It's not true everywhere, but it's definitely true in my group. I don't think I could speak to commercial fisheries stats just because I just started and I haven't met them all, but I get to work with a lot of really great men and women.
So Emily, this is Lisa behind the scenes. One of our social scientists at our center has actually done a study on the participation of women in commercial fisheries. So if you're interested, you can just Google women in commercial fisheries NOAA, and it should bring up the article that talks about the study and the contributions of women in commercial fisheries in Alaska. And I would imagine that I think that the study was also enlarged to include fisheries around the world too, but I'd have to go back and check.
Oh, that's awesome. Thanks for directing us all that way. And I would say, you know, my group has seen a lot of change over the past couple of years, just with retirements and new hires. I don't think I could say that it was as even as it is today. I'm really glad to be part of that change.
Awesome. And there still are a few more questions, but I'm going to let you move on and we'll get back to the questions after.
Awesome. Sounds great. So, you know, being on the boat, I kind of was getting to this. It's a huge team effort. That's my colleague, Rebecca. We've got this big haul of fish and we got to go through. It's a beautiful day and there's some amazing fish here, so we're all taking pictures. But it's a team effort and that couldn't be more important. So, you know, you guys have probably, I think previous talk has been given by Suzanne McDermott, and she worked with folks on St. Lawrence Island to do this cod tagging project. You know, Suzanne can't be everywhere, so we help out the team. And so here we are at Cynthia and Rebecca here, and we're putting in these tags. It's sort of like getting an earring, so it's not like super painful or anything. But we'll put these tags in. They'll be in for 30, 60, 90 days, sometimes longer. And you can see that we're learning a lot about where these specific cod go. And there's a map in the lower right-hand corner that shows some of where they're headed to and where they're going from where we measured them. So yeah, some really cool research. And you know, this is a special project. We're all in it together. We want to know what's going on, and many hands make light work. So for the folks who asked about how we tell how old a fish is, so on the boat, we might have, here's a Pacific cod. Right about there is where this this ear bone, this otolith is. On the lower, on the right side is a Greenland turbot otolith. And I put that there because I think they're really, really beautiful. But you can see in the middle picture, there's actually a wide array of what these look like. And you can see these rings, right? So I think I do that in the next, yeah, right. I made this up because I can't actually see the rings on this particular one, but you know, if you see the rings on there, you have to polish it. There's also all this processing that goes in, but you can start to see these rings and estimate how old these critters might be. In the lower left-hand corner, we put them into these boxes in these little vials, and we send them to the age and growth team. And so they're the ones who, while we collect those otoliths for them, they're the ones who actually look and see how old these critters are. So I put a little question here. These rings on this otolith, does that remind you of anything?
It looks like Theodore in the chat says fingerprints. Mabel
Okay.
and are thinking tree rings.
Yeah,
I think we have some smart viewers on. I think we have some smart viewers on because they saw one of our webinars on otoliths last year, and they are remembering all of this stuff. So that's great.
Oh, that is so exciting. Yeah, and it's just like tree rings. So each ring is made by like, well, to be less technical, as the seasons change, they're eating differently, depositing and making that otolith bigger each time. So there are these rings, and it's just like a tree. A tree would do that too. So you'll see all these tree rings, and it's really change of the seasons, but seasons happen once a year really, and so we get to see how old these amazing critters are. And so, you know, we do all this incredible research, we get to take these samples, but it's also a team effort on the computer, right? Like we're collecting this data, and so how do we collect that? So we have these tablets, that's the tablet. We're taking some notes about what we caught, how we sampled that. You see sub and non-sub, that's sub sample and non-sub sample. Going back to that population idea, how we're understanding the population and what we caught and how we're sampling that. And then we put it on the computer, and so we have this system that we enter all that data into. And when we get back to land, we use that to create some really important reports and documents. Earlier this year, you guys may have seen the Northern Bering Sea groundfish and crab trawl survey highlights. That was geared towards folks in the Northern Bering Sea region. If you want a copy of that, I think our comms team can provide that to you. And then we also made a presentation at the University of Alaska Fairbanks Straight Science Seminar Series. And I want to give a big shout out to them for letting us share this incredible work about your backyard. One thing that I have a personal goal of in this position is I want to share my science, and I want to be transparent about my science. So if you guys want to see the kinds of stuff that I'm up to, you actually can. There's this incredible place, online website called GitHub. And so I have an account on there. I share a lot of how I'm working through data and different data products to make better science for you guys. That's like the whole point. And so if you're interested, we have these repositories. I write a lot of code for my job. You can actually go and take a peek at what some of this looks like there if you're interested. I talked a little bit about Straight Science. They have an amazing webinar series. So you can learn about the science that's being done in your community, in the backyard, in Bering Strait region, elsewhere across Alaska. There's some really great work being done. And so, yeah, I can't highly recommend this seminar series enough. And then, you know, this past year was a little difficult with COVID, but in other years we've had some wonderful events that have been a great way to meet you guys and learn from you. And so we have, and I would say I want to give a lot of props to our comms team for organizing this. But seeing these fish firsthand, working with them, understanding what how they've adapted to this environment, and other really amazing hands-on activities. And so want to give a huge shout out to these opportunities in the future, and that you guys can get involved too. Students specifically, there's a whole website dedicated to student opportunities, and this is on NOAA's webpage. Actually, I have the link at the bottom, and so it has information about internships and boat experiences, outreach events, so much more. You know, it's not fisheries related, but the GOA 1617 satellite is being launched today. So yeah, I'm hoping to tune into that and see a rocket get launched. It's just so exciting. So anyway, there are a lot of great opportunities, and you can find them at this site. And then as promised, right, here are a bunch of people that I get to work with, these amazing scientists, and we all contribute to this incredible effort together in really different ways. And it couldn't be done just by one person. So team effort is integral to our work. You've seen some of these photos and some of these people and other photos from this presentation. If you haven't, I need to get more photos of them to add. But it's an incredible group of people, and I'm really honored and so glad that I can be part of it, and part of this incredible effort. I saw in Lisa's other talks that she asks for this, you know, if I had to give a piece of advice, I have a lot of thoughts on that, but, you know, I think you really, you know, when the going gets you down and you're working really hard, you just got to keep your eye on the prize. And so I've got pictures of me here, and you saw this picture earlier of me at the aquarium, looking at these fish in this aquaria. Also, I found this amazing picture of me. I was just home of me on the computer as a kid, and how far have I come from then? And if I, you know, go back to that moment, I don't think little Emily would have ever seen me here. But I worked really hard and steadfastly to get here, and so you just got to keep going for it. And I think, you know, making mistakes along the way just means you're trying. So make mistakes, because that's the only way you get through it. If I did everything perfectly, I probably wouldn't have learned anything, and I've learned a lot from my mistakes. I'm very dyslexic. Maybe some of you are too. Do you know what that is? You switch your letters and numbers around and get all sort of, writing is very hard, reading can be quite difficult. But I was so enthralled by the ocean and seeing what was there, and really wanted to study that, and so kept my eye on the prize. And just a huge thank you to all of you. It's a true privilege to study and learn from this ecosystem and from all of you, and I really look forward to meeting you guys maybe one day in the future in person. So yeah, I'm sure you all have more questions, and hopefully, you know, right on time, but I can stay on.
Awesome. Thank you so much, Emily. And we do have some more questions, but unfortunately we are out of time. We're going to take a few weeks break on our NOAA webinars because of March Madness and the Iditarod, so go and enjoy those events, and we'll come back at the end of the month with another webinar on March 29th. Thank you everyone for coming and your awesome questions.